So this is kind of an intermediate step right before glycolysis. This is formation of something called acetyl or acetyl-CoA. And I kind of look at this more like a messenger who brings the pyruvate, or what's left of it, to the Krebs cycle. So it's kind of like a taxi. I look at it that way. So here we are. And there's the pyruvate. It's going now into the mitochondria. And we can see the first thing that's going to happen here soon is that this little pyruvate is going to, look at that. What it did now, you can see here, is that one of the carbons with two of the oxygens, because remember, even though this just looks like two beads of carbon, it still has hydrogen ions or hydrogen atoms and oxygen atoms attached to it. So one of these CO2s is going to pop off, and this is going to leave the mitochondria, leave the cell, go back to the blood vessel, hop on an erythrocyte, and you're going to breathe it out. And so there we are, we got rid of one of them. So this is called now acetyl or an acetyl group, a C2 group. So this poor little acetyl group, oh, look at that, there's choo-choo train, we're stealing some more electrons again. This is coenzyme A. So the whole molecule is called acetyl coenzyme A. And this little blob just basically brings this acetyl group over to the Krebs cycle. So we're going to see it do that. And now here's the Krebs cycle. And this is a very complex system. But what I'm basically going to do is just kind of give you an overview. I do not care that you understand all the ins and outs of it. But basically what's really happening is, let me go to right before it, boom. You're going to see here that there's a 4-carbon molecule here called oxaloacetate. I don't even care that you know the name of that. But the idea is you have 1, 2, 3, 4-carbon molecule and a 2-carbon molecule. And let's kind of see what happens. So the coenzyme A brings over the acetyl group, which is these two. And now it has 6 carbons. So what's going to happen now, at some point, oh, one of the carbon dioxide, notice one of the CO2s from the acetyl group popped off. So there's a carbon dioxide. Here comes the choo-choo train again to steal some more electrons. And you're going to note that, oh, there's another one. So you could argue that these two things right here used to be joined together. They used to be the acetyl group. And now they're basically going to be sent out as trash. So what we're looking at here is from that one glucose that was split in half, it became pyruvate. One of the CO2s was removed before the Krebs cycle started, and now the other two. So literally there's nothing left from the original glucose molecule, because if you remember, the other pyruvate is doing the same thing. So there literally is no more carbon left over and no more oxygen left over from the original glucose. Literally, Krebs cycle just dismantles whatever is left from the original sugar molecule. This is where the choo-choo trains come into play. So let's watch them a little bit, because what we haven't talked about is this. As we know, glucose is C6H12O6. We can account for the carbons, we can account for the oxygens, we haven't even talked about the H's yet. This is an electron and proton carrier. They're called NAD and FAD. And basically I call them little choo-choo trains, and they carry coal, C-O-A-L. And the coal is the electrons and protons from the sugar. And so really, the way I look at it is that's really the major part of this thing, is we need to extract the coal from the sugar to run a whole system called electron transport chain to make ATP. And that's kind of where the next part of the video is going to go. Oh, by the way, so we've made 1 ATP per pyruvate, we've made 1 ATP per Krebs cycle. So since there were 2 Krebs cycles, 1 for each pyruvate, the entire process of the Krebs cycle produces 2 ATPs. Now this little train right here, he's not NAD, he's called FAD, and he's just another choo-choo train, but he can carry 2 electrons instead of 1. And so we're going to see him do his thing there. So he's going to go over and fill up the, there he goes, and now he's filled, and here comes an NAD, and it's going to grab some electrons, and they are all going to head to the last stage of cell respiration. Of course, there it is repeating again. And that is basically the electron transport chain.